Next up, we have Erika Raven, who will be telling us how fetal neurotropic infection results in long-term disruptions of brain structure and function.
Great, thank you so much, and thank you everyone for being here. I have nothing to disclose. So primate brain development is a protracted process across the lifespan that can be shaped by genetics, environmental influences, and adaptive learning. However, in early life, we see that the most rapid and fundamental changes during so-called critical periods. And myelination is a big driver of this early maturation and follows a strict, regulated, and time-dependent program where different brain regions mature at different timings and rates. And importantly, the order of this maturation process really matters. So for example, brain regions and their connections that serve primary or basic functions mature first. And this kind of makes sense. So the first thing you need to do is to be able to sense your environment, and then you go on learning to walk and talk, and not much long after that, you can do complex integrative processes like learning to drive or maybe navigating your first OHBM conference. And in terms of basic functions, touch is the earliest sensory process to develop, and we observe that near the end of the second trimester. And so while the importance of brain maturation in early life cannot be overstated, it can of course be disrupted with profound implications for later life. And it is this disruption we think to define better over the course of the study. So we can now turn to a real-world example of early life disruption through the Zika virus infection. And so Zika virus is one of the few pathogens that can access the fetus during pregnancy, as it can cross the placental barrier. And this virus has been linked to a large range of disease phenotypes, notably impacting the brain. And the severity of these outcomes are heavily influenced by developmental timing. So first trimester infections have been linked to more extreme phenotypes, such as fetal loss, and devastating neurological consequences, such as microcephaly, where infants are born with abnormally small brain sizes. And in contrast, infections occurring in the second and third trimesters often result in infants with normal-appearing head sizes, but an elevated risk of developmental delay. So we now ask, what are the long-term consequences of disruption during critical windows of development? And so we can turn to the neurobiology. So what does Zika actually target? And so there have now been several studies examining the neurobiological targets, and we know that all cells of the central nervous system are impacted. But in particular, myelin, the axons that they myelinate are most vulnerable. And this infection time window is very fast. It lasts only about 2 weeks, which limits the damage to the most active processes during that time period. So overall, we can work under the premise that Zika-induced myelin loss may cause secondary neuronal damage with long-lasting consequences to brain development. So in order to study this and to better understand and model these early life disruptions, we can turn to the use of a common neuroscientific animal model, the rhesus macaque. Rhesus macaques have similar early developmental trajectories as compared to humans, in particular how their brains develop. So what we can do using these non-human primates is to investigate the relationship of both structural and functional maturation in the brain. And so we can then ask our first hypothesis, which is, do specific targets in time lead to focal brain damage, in particular regions undergoing rapid maturation? And we can even extend that to say, in particular, we would predict early sensory and motor regions to be preferentially impacted. And then we have a secondary hypothesis in that damage is irreversible, so we can actually observe this over time, later in developmental windows. So this schematic highlights our study design, which includes an age and sex-matched cohort of 9 control subjects and 10 Zika exposed. So the mother and fetus macaques were inoculated at the start of the second trimester with Zika virus to ensure exposure. And there were 4 time points of MRI scans, where scans were required at regular intervals to study longitudinal trends over time. And these scans were required at 6, 12, 18, and 24 months. And so these time points reflect an approximate developmental range of 4 to 8 years of age in relationship to human development. And so our main primary goal is to essentially look at the outcomes. So what happens after this early life perturbation? And so this is just an overview of our imaging and analysis workflow. So the non-human primate is actually large enough to go into a clinical scanner. So all of these animals were scanned at a 3 Tesla MRI machine. And the animals do need to be anesthetized to undergo the scan, so that means that they're asleep in the scanner, making motion artifacts and things related to that very minimal. They're also in a stereotaxic frame, so we can align the scan series within session and over time for the subsequent visits. We then had a multiparametric acquisition where we acquired data with similar SNR and resolution as it relates to human scans, and the sensitivities of our contrast-based measures were tuned towards myelination and tissue microstructure. There was then quite extensive pre-processing that needed to go into all of these different contrast mechanisms, but we were able to do that. Some of these include intensity normalization and ultimately alignments to templates and atlases. And that allows for feature extraction, where we can then look at the size and shape of the cortex, but also these contrast-based features. And in addition to this, we also had a task-based fMRI paradigm to look at neural processing. And then finally, we can model some effects. And so in early developmental studies, it is critically important to be able to disentangle age versus the effect of interest here, which is disease. And so we were able to use linear mixed effects modeling to this extent. And then, so to start off with our structural findings, and so we're looking for unique patterns of effects across a variety of features. And so here I have a cortical atlas showing the 4 lobes of the brain in 4 different colors. And we have different features related to macrostructure. So these include thickness, surface area, and volume, and also microstructure. And so as I mentioned before, we tuned our parameters to be sensitive to myelination properties and also microstructure. So this could be anisotropy or alignment of tissue. And then, so these figures illustrate the significant correlations between pairs of parameters across all the regions to identify signatures of metrics that might be specific to either the age or disease status of the animals. And so just looking at this, we found quite a lot of correlations across age, but age is very strong. These animals are really undergoing a significant maturation process in both cohorts. So what we really want to dive in for the point of the talk today is like, what did we notice in terms of the impact of Zika? So just to start off with some MRI signatures, so what we ultimately observed is that the impact of Zika was restricted to a discrete set of regions within the parietal and frontal lobes of the brain. And the specific MRI features that were associated with these effects represented what we would call an immature phenotype. So these include decreases in surface area and myelin-sensitive MRI metrics, and increases in cortical thickness. But interestingly, we did observe an anomaly in this feature set, and this is with increases in a diffusion metric of fractional anisotropy. So typically, you would expect fractional anisotropy to go up with development, and this has been shown many times over the years in both cross-sectional and longitudinal studies, in humans, in animal models, and so we would have expected the opposite trend in the context of a disease group, where you would say, oh, my FA will go down. That's not what we saw. However, in past studies of congenital disorders, and here I'm referencing 22q11 deletion syndrome in these references below, we and others have observed this unexpected increase, which goes along not just with the measure of FA, but also restricted or smaller volumes, potentially indicating less space, leading to increases in anisotropy. So less space is available, but ultimately the fibers are still organized and densely packed. And then if we go on to examine regional specificity, so the strongest effects were observed in our primary somatosensory and motor regions and seem to extend out into the surrounding regions. Okay, and so several of these regions also belong to networks and processing that support discriminative or effective touch. And so we can go on to probe that in our next experiment, which looked at the neural pathways of touch. And so touch pathways are conserved in both humans and in monkeys, and alterations in somatosensory pathways have been reported as being disrupted in neurodevelopmental conditions such as autism disorder. And so the task that we used for this was adapted to the macaque. It's a task-based functional MRI paradigm, and it was originally performed in adult animals. And so there were 3 conditions: one for effective touch, which is essentially a pleasant touch, a gentle touch; a discriminative touch, which is how you perceive your environment; and then also a rest condition. And so looking at the main regions involved in the cortical network that we can investigate, We are showing first that we were able to replicate the results from the adult animals, which is important since we're translating this to a developmental study. That the slow touch resulted in significantly increased activation across all of the regions. But in particular, for the Zika insult, we observed that the primary somatosensory cortex had a significant interaction between touch speed and group, where Zika had decreased response activation for both conditions. And so bringing all together our structure and functional results, we observed that perturbation during the second trimester impacted long-term maturation within a discrete set of frontal and parietal brain regions. And the greatest impact was seen in the primary somatosensory cortex, which also altered touch-based activation patterns. And these results aligned with the timing of perturbation, histological evidence of myelin onset, and preclinical evidence of myelin damage due to Zika infection. So I now want to thank everybody that contributed to this project and also the funding that supported it. And I am available at poster 303 today if you want to chat. Thank you.
What a fascinating talk, thank you. So we have a question here asking, are the infected animals kept for studying effects over the course of their life?
So the imaging that was done for this was part of a larger study, and so the main end goal of the study was to look at the histology. And so other investigators involved are looking at basically what underlies the MRI features that we can observe to understand like exactly what cell types, what local connections might be perturbed. And so that is also underway.
Thanks. Any questions from in-person audience? You can, yeah.
How do you establish the causality between the Zika virus infection and the histological changes?
So this is a perturbation, so technically we're not looking directly at causality. So this is just looking at the impact over time. However, is part of a sort of subsequent investigation, there will be histological measures available as part of this project, not analyzed by me, but by collaboration partners. And so they will actually be going in to look to see, okay, what are the long-lasting consequences of this infection? Because we know from some of the histology that I was showing at the beginning, like, yes, you can see that the myelin is decompacted. You can look at, like, genetic signatures. You can look at different protein signatures. Like, everything points to the myelin in terms of the immediate insult, but then in terms of how cell types might recover or how they mature long-term is, you know, we're looking at this indirectly using imaging. And so causality then becomes difficult to establish. And so hopefully that histology investigation will then show us, like, in particular for those sort of unusual results that we saw with the fractional anisotropy going up. Like maybe we see some differences in terms of packing and density of local fibers in those areas. So TBD.
So it's like if we just skip the infection part, at least we know that the myelin part is already like have observable changes.
Yes.
Okay, we do have time for one more question.
Sure.
I was curious about, you selected the beginning of the second trimester, and what would you expect regarding global behavior of the animals? Did you see any global behavior?
Yeah, so that's a good question. So the timing of the infection was very early on, and that was kind of intentionally so because they were trying to find the sweet spot for the model where the ultimate goal was to kind of observe for infants that were born with typical head size, could we observe any deficits long-term that might be sort of under the surface? Because as you might remember from the news in 2016, the extreme phenotype of Zika was very devastating in terms of microcephaly. So yeah, so that was kind of one of the reasons for choosing that timing of the model. And behaviorally, so the animals were assessed multiple times over their lifespan for any motor deficits, any visual deficits, interaction deficits, and interestingly, the main thing that came out of this were emotional deficits, so the pair bonding relationship between the mother and the infant. And yeah, I guess I'm not sure how We did not use those data in relation to this imaging study. We were looking more at like processing and structure and neurobiology, but it would be interesting to kind of understand if there's a brain basis, but for those, but I'm not sure.
All right, let's thank Erika again.